panelists. Now I'd like to turn it over to our Chief Scientific Officer, Professor Amin Rostami, who will introduce today's speakers, Niloufar Marsousi and Jeannine McCune.
Thank you very much, Susan. It gives me great pleasure to introduce our speakers for today's webinar. The two speakers that we are having, it's truly a double treat for all the audience. They are going to present, while you are reading their, the short resume, they are going to give two different angles to the applications of modeling and simulation in healthcare. I have been fortunate to know personally both of the presenters today. Dr. Niloufar Marsousi has been working in Geneva University Hospital and is going to show cases that involve what we so-called, bottom-up modeling of putting the information about the patients and seeing that, what is the best dose that we can prescribe, and the focus would be on the DI side, while Dr. Jeannine McCune is going to talk about a different approach to modeling and simulation, which involves actually Bayesian adjustment on the back of some measurements that we are having and the models that they have been developed generally for that particular compound. If Susan allows me, I am going to have a couple of intro slides that why we are talking about this. If we actually look back what Certara's interest is in this area, I have to take you back to May of last year when Certara sponsored to have a meeting with regard to the modeling and simulation application in healthcare, which happened in Manchester. This meeting was basically a self-critical assessment of why modeling and simulation has not become the norm in clinical practice, whether there is a problem with the technique itself or there is something else. So everybody who came to that meeting, they had cases of modeling and simulation, and hopefully this is what you will get from the presentations today, to indicate that the modeling and simulation in healthcare not only, is used, in many of these, I would say, pockets of the excellence in different hospitals and different settings, but it has been very useful. And in the next stage, basically what we asked the presenters and attendees to do is to highlight what then are the problems. If the scientific problems are not the issue, what has stopped these models to become common? And I am glad to let you know that as a result of the contributions that many of those key opinion leaders had, we had a white paper which is now accepted for publication in CPT. Both Niloufar and Jeannine have been co-authors on the paper that you are seeing that is going to come out, where as you can see in the title, the results from those debate and discussions outlined, and as I indicated, they are in line of why this particular, let's say, different kinds of modeling has not picked up. It talks about the variation in the utility as a result of the covariates that we are having, different techniques. It summarizes that the models that you are going to see, the two sides of it today, from Niloufar's side as well as Jeannine's side. And then it brings up the elements that makes these modeling and simulation much more wanted in certain area, and whether these have been fulfilled with the previous examples or not. And eventually, it actually brings up a futuristic view of how these can be integrated to a level that application of modeling and simulation becomes common in every clinical setting. And it talks about different tools, you know, the interfaces, education, many aspects. So I will invite for all those interested audience for today's webinar that to keep an eye on this paper. It's going to appear in the May issue of CPT, but with that introduction, I would like now to invite our first presenter, Dr. Marsousi, to present her case and how actually they got into it, you know, how they did it and what the results were, etc. And hopefully you will get to the same conclusions that we had in the paper with regard to how these techniques can become more common, knowing that they are actually doing a good job. Okay, with that, Suzanne, I would like to pass the presenter rights, or however this is working, to Niloufar. Okay.
Hello, everyone. Thank you, Amin. I am happy to present you the study we've been working on at Geneva University Hospitals, which consisted on a case of dose adjustment for co-administration of ticagrelor and ritonavir. As you may all know, there has been a significant progress in development of antiretroviral treatment for treatment of HIV infection, which means that these patients are having an increased life expectancy with a mortality rate which is close to uninfected people. So just by the fact that these patients are getting older and by the nature of their pathology and the side effects of the treatments, these patients are at a higher risk of developing cardiovascular problems compared to uninfected people. For example, there's been some studies that showed that the annual relative risk of cardiovascular events in HIV patients is more than, it can be between 1.26 and 1.75 compared to uninfected people. So an appropriate cardiovascular prevention is really required in more than half of these HIV patients, and this proportion can increase in the future. So just as a reminder, I'm sure all of you know that ritonavir is a protease inhibitor drug which is almost present in any HIV treatment, and it's a very potent inhibitor of cytochrome 3A4 and PGP transporter. This compound is used basically as a pharmacokinetic booster because it inhibits the elimination pathway of other antiretroviral treatments and increases their bioavailability. So in a frontline therapy and treatment of cardiovascular diseases consist of mainly antiplatelet drugs. We have two main categories of antiplatelet drugs. We have irreversible drugs like clopidogrel and prasugrel, and reversible ones like ticagrelor, which I will talk about later. So in terms of clopidogrel and prasugrel, it's important to know that these are prodrugs, which means that they need to be bioactivated via different cytochromes. Clopidogrel is bioactivated via cytochrome 3A4 and 2C19 and some other isoenzymes. It's very well documented that the response to clopidogrel is very variable, and we can have up to 35% of patients taking this drug that are non-responders. On the other hand, we have prasugrel, which needs to be bioactivated via CYP3A4 and 2B6. And so it's clear that since they are prodrugs, if we have a drug that inhibits their bioactivation pathway, the real problem is an inefficacy of these compounds, which is what we observe in HIV patients. On the other hand, we have ticagrelor, which is the reversible antiplatelet agent, and it's active itself, and it also produces an active metabolite via cytochrome CYP3A4. So in case of co-administration of ticagrelor and ritonavir, the problem basically is the other way around, which means that we are having a bleeding risk because the patients can have accumulation of ticagrelor in the body. So since it's known that the pharmacokinetic of ticagrelor is linear up to 900 milligram daily doses, we just imagine that if we reduce the dose of ticagrelor proportionally to the observed AUC ratio of ritonavir-ticagrelor drug-drug interaction, we may just be able to counterbalance this interaction and avoid this bleeding risk. So we conducted a clinical study which was based on a bioequivalence clinical study in healthy male volunteers. At the first session of the study, we administered ticagrelor 180 milligram single dose, which is the loading dose really used in clinical practice in patients, and we assessed the pharmacokinetic for between 24 and 48 hours. And we assessed the platelet inhibition in volunteers using two gold standard platelet reactivity tests, which are VASP and Verify Now. And also we phenotyped these volunteers and we assessed the activity of CYP3A4 and PGP using very low doses of probe substrates, midazolam, and fexofenadine. During the washout period, we simulated the interaction between ticagrelor and single dose of ritonavir, and based on the obtained AUC ratio, we calculated a new dose of ticagrelor to be co-administered with ritonavir at the second session of the study. And the second session was just conducted the same way as the first session. So ideally, we wanted to have exactly the same PK profile and also the same platelet inhibition at both sessions of the study, avoiding all side effects, overdose, or bleedings in patients, in volunteers. So we built PBPK model for ticagrelor and active metabolite based on published in vitro and in vivo studies, and also some in-house data. For example, we generated information about permeability of ticagrelor using KECO2 cell lines. Once the models were built, we refined them based on a published clinical study where the PK profile of two compounds were assessed in healthy volunteers. And at the end, we validated the model based on another clinical study where the interaction between ticagrelor and ketoconazole was assessed in healthy male volunteers. We just compared the AUC ratios we obtained with our models and the AUC ratio observed in this clinical study, and we could validate the model. Once the model was validated, we simulated the interaction between ticagrelor and ritonavir single doses in 20 virtual healthy male volunteers, and we obtained an AUC ratio of 4, so we just reduced the dose of ticagrelor by 4-fold and administered this dose at the second session. Here are results in terms of pharmacokinetics of ticagrelor and its active metabolite. As you can see, the AUC values of ticagrelor at both sessions were very comparable, even though we administered 4-fold lower dose of ticagrelor, and the formation of the active metabolite was almost completely inhibited. This is what we expected. In terms of CYP3A4 activity, as we expected, the activity was almost completely inhibited by ritonavir. But what was really surprising for us was that we observed that the PK profile of fexofenadine was exactly the same at both sessions of the study, which means that ritonavir did not really have any impact on pharmacokinetics of fexofenadine. Here are observed and simulated data that are overlapped, that you can see that there were a good correlation between concentrations that we could simulate with our model and observed data. The dashed line represents the simulated data with ritonavir, and continuous line represents the simulated data without ritonavir, and dots represent observed concentrations. In terms of pharmacodynamics and platelet inhibition, as I said, we used two different platelet activity tests, which were VASP and Verify Now. VASP assay gives the results as PRI values, which is platelet reactivity index, And so based on different guidelines, a cutoff value of 50% is defined, which means that if a patient takes antiplatelet agents and has a PRI value of higher than 50%, it can be classified as a non-responder. On the other hand, we had Verify Now test, which gives the results as PRU values, which is platelet reactivity units, and the same way the defined cutoff value is about 206. So we observed that, first of all our subjects had a platelet activity below the cutoff values at both sessions of the study, which means that even though we administered a lower dose of ticagrelor, we could still obtain the desired platelet inhibition by ticagrelor. And most of all, we did not have any overdose or any side effects. We did not observe any bleeding in the study. So as a conclusion, this dose reduction based on modeling and simulation allowed us to maintain the optimal platelet inhibition in both sessions of the study without any side effects or bleedings. It is important to know that to be able to broadly use of this model, we have to do more validations at steady states of both compounds. It is very important to know that ritonavir is known to be a mechanism-based inhibitor of CYP3A4 and also inducer of some other different cytochromes. This can only be assessed in a steady state level. And on the other side, it will be very good to be able to incorporate special physiopathological parameters with regard to HIV population. If we have other drugs, if we have modulation of different cytochrome activity or protein bindings, all these parameters to be included in the model. And also, it's something to do that we want to do the PD modeling of the activity of ticagrelor. At the moment, we are running another clinical study where we focus on the other category of antiplatelet agents, which are irreversible ones, clopidogrel and prasugrel. In this study, we have two groups, HIV patients with a boosted therapy, which means that they have a cobicistat or ritonavir as a pharmacokinetic booster. And on the other arm, we have healthy volunteers taking single dose of clopidogrel and prasugrel, and we want to, at the beginning, just assess the difference between PK and PD of both groups and also try to validate the model and do PBPK modeling with this information. I want to thank you for your attention, and if you need to see more details about this study, I invite you to see our publication, which was published on CPT in June 2016, and I will be happy to take any questions. Thank you very much.
Thank you so much, Niloufar. We'd like to invite our audience to submit their questions to Dr. Marsousi through the Q&A panel. Nilo, it looks like we've got a question from our audience. Someone would like to know if ticagrelor is a substrate of OATP 1B1 or 1B3, as ritonavir inhibits this transporter?
This is a very good question. Unfortunately, there are no data about this for ticagrelor. This is what we try to find out, but there are no information. So we suppose that it's not a substrate of these uptake transporters.
Someone in our audience would like to know, given the interaction problem regarding both reversible and irreversible platelet inhibitor or platelet agents, what is the current clinical practice for cardiovascular disease prevention in HIV-positive patients?
This is also a real problematic here, at least at Geneva University Hospitals, because everyone knows that ticagrelor has a higher efficacy compared to other two compounds, but in the notice of manufacture, at least at Switzerland, it's clearly said that ticagrelor and ritonavir are contraindicated. So, and in clinics, they are not prescribed at all together. So clinicians, what they do is just they try to find an alternative, but very, very commonly it's clopidogrel that is prescribed. And they are all aware that it's not working because they are having a lot of cardiovascular events, even though patients are really taking their clopidogrel pills, but this is what is done for now.
What are some potential reasons why you didn't see any difference in pharmacokinetics of fexofenadine was observed when it was co-administered with ritonavir?
One of them, I think the most important one is even though fexofenadine is very commonly used as a probe substrate of PGP activity, we know that it's a lot of other transporters are involved in the transport of these compounds. For example, exactly just the other participants said the alloHCT or alloHCT 1B1 and 1B2 and even other transporters. So the main reason we think that one is that ritonavir interfere with other uptake transporters, and the other thing might be that ticagrelor also might be an inhibitor of a transporter, for example, MRP2.
Looks like we've got one final question from our audience for Niloufar. Why was the dose of 180 milligrams of ticagrelor chosen for your study?
Simply because it's the common dose used in clinics as a loading dose. Patients start the treatment by taking a single dose of 180, and they go on by 90 milligram once a day. So that was just the reason.
Perfect. Thanks, Nilo. I'll now turn it over to Jeannine for her presentation.
Thank you, Suzanne, and I'd like to thank Suzanne and her colleagues at Certara for inviting me to present today, and Niloufar for a great presentation so far. So today, I'm going to talk about model-guided selection and dosing in allogeneic hematopoietic cell transplant patients. And this is work that I've done as a professor at the University of Washington School of Pharmacy and also a member at the Fred Hutchinson Cancer Research Center. So in terms of the context of precision medicine and allogeneic hematopoietic cell transplant, abbreviated alloHCT, this is a procedure that's used for patients with inherited disorders or refractory cancers. And although malignancy is the most common indication for receiving this procedure, children with inherited disorders such as SCIDs or severe combined immunodeficiency and other genetic conditions often receive this. So allotransplant has a really unique patient population because it covers the whole age spectrum, to infants to elderly. And the goal with doing an allotransplant is that you want to cure the patient of their underlying disease with no regimen-related toxicity or no graft versus host disease. And graft versus host disease is unique to allotransplant, in which the donor graft, which the hematopoietic cells had been infused into the recipient, the allogeneic donor cells or the graft then attack the recipient or the host. And so that's GVHD. And allotransplant actually got its start here in Seattle, and it really started on the premise of dose intensity, where we would give very high doses of older, and the chemotherapy that was given was off-label, and that's still true today, with substantive and historically it was fatal toxicity. So the table at the bottom of the slide shows the usual timeline of an allotransplant, where everything is relevant to day 0, graft infusion shown here. And the majority of my talk today will focus on the conditioning regimen, where you are preparing the recipient for receiving their graft, although there's a lot of opportunities for model-based dosing in post-graft immunosuppression. I won't go into that for the sake of time. Now, one unique feature to allotransplant in the context of overall oncology is that although it is used often, allotransplant is too rarely used to do a randomized controlled trial of when we talk about conventional versus PK-guided dosing. And taking a step back and looking at oncology in general, at least in the United States, any dose personalization method that is used in oncology has to improve the efficacy of the chemotherapy regimen or avoid lethal toxicity. toxicity. And because of a lot of the advancements in drug development that have been made over the past 20 to 30 years, we don't have to approve drugs that have frequent lethal toxicity. So essentially in oncology today, PK-guided dosing needs to improve the efficacy. In oncology, just reducing manageable toxicity is not enough. And the table here below shows, in the context of oncology, that there's only been 3 trials that I know of that have studied conventional dosing to PK-guided dosing, and none of them are in allotransplant. So because of this limitation with the number of patients that are available in Seattle, our general approach has been when we're trying to answer the question, does PK-guided dosing improve outcomes? It follows this general algorithm. Since all of our drugs are off-label and they're very old, really historic data doesn't really help us because we have such newer techniques now, not only in terms of LC mass spec, but also in terms of modeling. So my group has developed usually novel analytical methods where we characterize the pharmacokinetic variability of the parent drug and the metabolites, and then we conducted a pharmacodynamic study to determine if the AUC or the trough concentration is associated with clinical outcomes in a homogeneous population. Now, one challenge with allotransplant is even with big transplant centers, you end up having some diversity in terms of your patient population, which can obscure a pharmacodynamic observation. And if we do see an association between an area under the curve and clinical outcomes, we will then what we coin a phase 2 study where we will evaluate the benefit of a PK-guided dosing in and compare it to historical controls. And then if we see a clinical benefit, we then have clinical acceptance of precision medicine with PK-guided dosing. And today, where you will see PK-guided dosing is with the alkylating agent busulfan and then the calcineurin inhibitor cyclosporin and tacrolimus. Now, really over the past decade, we've had a revolution in terms of personalized medicine. And I think one of the best examples is Watson and how there was recent data that showed that using the computer algorithms that were created with in collaboration between IBM and Memorial Sloan Kettering, that basically computer-guided chemotherapy selection can help oncologists with choosing the chemotherapy regimen. And although this is a very different type of context than popPK modeling, I think this shows us that the technology is moving really fast and that, you know, really the concept of pharmacokinetic guided dosing has come from the 1970s with Shiner and Beal, and now we have the tools to move it forward. And I hope that the Watson example uses this as a call to move forward at a more rapid pace. So here is a series of studies that my group has done in collaboration with a number of wonderful pharmacometricians ranging from Paolo Vicini to Don Major, Jeff Barrett, and Nick Holford. And we have used popPK in a variety of research settings where we have used it to model drug-specific effects, such as the fludarabine-induced lymphodepletion of a conditioning regimen. And then we've also used it in the context of post-graft immunosuppression to model how mycophenolic acid concentrations relate to its target enzyme, inosine monophosphate dehydrogenase. We've also used popPK-based limited sampling schedules to understand the pharmacodynamic association, because there is a type of allotransplant that can be conducted in elderly patients called non-myeloablative transplant that is completely done in the ambulatory clinic, and those types of transplant need to have shortened sampling schedules such that we can allow the characterization of the pharmacokinetics and less the pharmacodynamic association. Also, we have done a study where we showed that you can lower liver and renal toxicity with popPK-guided dosing of cyclophosphamide, which is a prodrug, and we actually dosed the parent drug based on the target AUC of 2 metabolites, 4-hydroxycyclophosphamide, which is associated with rejection, and then carboxyethyl phosphoramide mustard, which is associated with liver toxicity. And in that study, what we did is we obtained real-time concentration time data from the patients, and then within the course of 5 hours, quantitated those samples using 2 LC mass spec methods, and then used a Bayesian R dose adjustment algorithm created by Paolo Vicini and Dave Salinger to do in real-time popPK-guided dosing of cyclophosphamide that was then used to personalize the drug dosing in patients that received a cyclophosphamide total body irradiation regimen. And also, I really feel that where we need to move popPK dosing is in terms of using that for busulfan. And I think there's a huge untapped potential of physiologically based, pharmacokinetic-based dosing of busulfan. And this is really in the clinical context. So this slide shows the acceptance of PK-guided dosing of busulfan. This is data in terms of the number of patients that had their areas under the curve quantitated for PK-guided dose personalization done at our Seattle lab that's solely dedicated to busulfan. We started dose personalization in 1996, and you can see by the darker gray bars that initially it was mainly patients treated here at the Fred Hutchinson Cancer Research Center. But there's 2 main trends that I'd like you to get from this slide. First of all, you can see that we basically now have 4-fold higher use of PK-guided dosing of busulfan than what we started with 20 years ago. And the other thing is that only a minority of patients now are Seattle patients, and the majority of them are outside patients, mainly within North America, where we personalize drug dosing using AUC for over 60 transplant centers. So what is the basis for PK-guided dosing of busulfan? And this is actually data that started in the late 80s and really evolved over the past 20 years. And busulfan AUC is associated with 3 clinical outcomes: rejection, which is when the recipient's body does not accept the donor graft, and this is a fatal event; liver toxicity; and then relapse in the disease CML. I'm not going to focus on the relapse in chronic myeloid leukemia data, other than to say that this was generated before imatinib and the targeted tyrosine kinase inhibitors were FDA approved, and that there's really a big need to better understand the association of busulfan AUC with relapse in other diseases. So really today what's driving the use of PK-guided dosing of busulfan is the rejection and hepatotoxicity. And you can see here that the pharmacodynamic association studies were really in small patient populations. And also when we or others did phase 2 studies, you can see that there were small patient populations that showed a dramatic benefit in terms of PK-guided dosing. So the way that targeted busulfan, as it's called in the allotransplant setting, and it's also really essentially PK-guided dosing or therapeutic drug monitoring, is conducted today, is that the first dose is based on body surface area or body weight. You get multiple blood samples to estimate the AUC. Then using the traditional clearance equals dose divided by AUC, you get the patient-specific clearance. And then you adjust the dose based on the patient's clearance to achieve their target AUC. And one thing that I have here at the bottom is showing the relationship between a busulfan CSS, or concentration at steady state, to AUC. One thing that's really been tough with communicating busulfan pharmacokinetic data to hematopoietic cell transplant physicians or transplanters has been that there's a different vocabulary. And I think this is one thing that we really all need to work on for precision dosing, is how do the different disciplines talk to each other. And we actually had to use the term CSS to help with the transplanters understanding the concept, although obviously AUC is a term that's much more accepted in the pharmacology world. So our group has done a variety of different approaches to try to improve PK-guided dosing of busulfan. With the current dosing methods, basically only a quarter of adults and children achieve their target AUC with weight-based dosing of busulfan. We've also taken the candidate gene approach, looking at the drug metabolizing enzymes that break down busulfan, with GST-A1 being the predominant enzyme and GST-M1 being the next one. And we have found that GST A1 and M1 genotypes do not adequately predict IV or oral busulfan clearance, such that dosing cannot be simplified using candidate gene approach. We also, in parallel, have taken a variety of different population pharmacokinetic models where we had sought to use a shorter sampling schedule to allow for outpatient PK-guided dosing of daily IV busulfan using post-Bayesian estimates. And this approach can really save the healthcare system a substantive amount of money because it could reduce the number of inpatient admissions. And then another approach that we have done is using our wealth of data, not only from our center, but from other centers, to create a popPK model where you can use one model to predict IV busulfan clearance using normal fat mass as a body metric, and that can help with initial dosing and hopefully PK-guided dosing. And we've built this model in children and adults, and we are currently validating it under the auspices of my National Cancer Center supported R01 to validate this in adults. And I think really there's an untapped potential of PBPK modeling, and although there is a PBPK model for busulfan that was created by Georg Hempel's group, there really is a need to develop models also to study busulfan drug interactions. So this slide just shows at a high-level view a variety of different population pharmacokinetic models. And really the bottom line I'd like you to get from this is there's a lot of consistency with these models. The covariates are showing that body weight is important. There is some differences in terms of what covariates have not been associated with the clearance, but essentially we have built good, solid models. And yes, it seems like there's a lot of work that could be done in modeling, but I really hope that we can take what models we have currently and while we're also refining these models, start reaching out to our clinician colleagues and making these models more accessible to help with patient dosing. So recently, the American Society of Bone Marrow Transplant Guidelines Committee came out with a considerations document. Because of the heterogeneity in the busulfan pharmacodynamic literature, we couldn't come up with guidelines, but they created these consideration documents that talked about personalized busulfan-based conditioning that mainly focused on PK-guided dosing. And in their guidelines, ASBMT stated that popPK models have great potential to improve the estimation of the preferred initial busulfan dose, abbreviated BU here, and an individual's busulfan clearance. So I think this is really a big moment in the context of the medical community is recognizing the need for popPK-guided dosing. And I really think that Jeff Barrett's group work that he did at Children's Hospital of Philadelphia with dashboards and the use of dashboards to make models user-friendly as shown in the gray boxes here with concentration time data with methotrexate can really be used to help with making these models accessible to patients. And I think there's really a lot of work that we can do there. So where I see clinical application of popPK models being is to focus on drugs that have PK-guided dosing that's clinically accepted. I talked mainly about busulfan, but there's also a lot of work that can be done with the calcineurin inhibitors. And they really have to be web-based and clinician-friendly with appropriate support. And so what's the advantage of these models? They can improve the efficacy, and I think we also need to take the approach that they can improve patient safety. And they also create a pathway for dose personalization and can lead to outpatient dosing with cost savings, as I showed with the shorter sampling schedules with busulfan, and they could also lead to fewer samples as well. But there are some challenges, at least in the United States, with implementation of model-guided dosing in that we can't let perfect be the enemy of good. We can't be focusing on building a better model while ignoring how we can get these models out in the hands of clinicians. And certainly funding is a struggle with this area, and we also need to work on improved communication between pharmacometricians and clinicians. And another thing is that we have to remember is that clinicians have insufficient training of pharmacokinetics, and this has been a trend that's happened over the past 20 years. And so we really need to make sure that we create a dialogue that is respectful but yet also clear on communicating our findings, and especially when you talk about popPK as well. And so actually, I'm leading with a variety of international colleagues a busulfan harmonization effort to try to improve the efficacy of PK-guided dosing of busulfan while also improving patient safety, where we're first going to hopefully create an international proficiency exercise where we will have regular worldwide checks of busulfan concentrations to see if data that you get in the United States, which the U.S. and Canada has a proficiency exercise, would those results compare over to Europe or Asia? The other aspect that we're doing is looking at the most reliable method to predict busulfan exposure, to estimate an individual's clearance, and then personalize their busulfan dose. And this is really where I think PK-guided dosing can really help with creating an infrastructure that we can have a dialogue for how do we move forward with either physiologically based PK dosing or popPK dosing. And then the other and last aspect is how to report busulfan exposure. And this slide just shows the different ways that busulfan exposure is currently being reported. Busulfan historically has been dosed every 6 hours, but now it's being dosed more often daily. And so there's differences when we use AUC, and we have to obviously talk about the context of the dosing frequency. Now, concentration steady state, when we talk about busulfan, is actually the AUC divided by the dosing interval. So its expression does not depend on the dosing frequency. But then when you compare what the U.S. does to what Europe does, Europe uses different units. And so this seems like an area ripe for a dosing error. And really, this is what's driving my desire to do this work, is to try to improve patient safety while also improving the efficacy here. Because this is very reminiscent of what was happening with allogeneic transplant when we were prescribing medications and there was an accidental accident that a patient received 4 days worth of cyclophosphamide in one day. And so I fear that something like that could happen here, so we're hoping to avoid that. So with that, I will take any questions, and I appreciate you all spending your time, and I'd also like to acknowledge the work that my laboratory had done along with my various and wonderful pharmacometric colleagues that I mentioned earlier.
Okay, thank you so much, Jeannine. We'd like to encourage our audience to submit their questions through the Q&A box to Jeannine. Someone has asked, how do you see the implementation of personalized dose manufacturing in today's pharmaceutical industry? In other words, how do we manufacture a personalized dose?
I think if you're talking about developing a new drug, I think that basically creating a model that has a web interface like the dashboard would need to happen in parallel with that. And I know that that can create a lot of regulatory issues, although I'm sure since I'm not in the private sector and pharmaceutical industry, I'm underappreciating the amount of regulatory work that can be done. But I think basically creating a cookbook as much as possible. There is actually in the package insert for IV busulfan based on also in the FDA and the EMA how to do PK-guided dosing, and that can be used as a starting point.
Someone would like to know, what pharmacokinetic analysis methods are clinically used currently?
So our group has reported what's often used, and currently today, non-compartmental analysis, which is really an older technique that obviously we can do much better than, is what's used most often clinically today.
What is the pharmacokinetic variability in busulfan clearance?
So busulfan, actually, when you look at it relative to other chemotherapy drugs, does not have a lot of pharmacokinetic variability. The interpatient variability in clearance is around 20 to 25%, and that compares it's very low, even relative to the drug imatinib, which is dosed on a milligram per day basis, which has a interpatient variability in clearance of around 40 to 50%. So I think this just speaks to the narrow therapeutic index of busulfan.
Looks like we have one last question from our audience. Someone would like to know what drug interactions are of concern in patients receiving busulfan.
So busulfan is a GST substrate. But yet it also has a lot of reported drug interactions with cytochrome P453A drugs such as itraconazole. So we worry about the azoles because these patients are immunosuppressed even when they're starting their conditioning regimen because of prior chemotherapy or their underlying disease. So the azoles are a big concern, as is the concomitant anti-epileptic drugs that must be administered to avoid busulfan-induced seizures. So the topic of drug interactions with busulfan is certainly an area ripe for research.
Okay. Thanks, Jeannine. And